Okay. Hello. Um, I'm recording this early, so you guys can listen to the recording instead of watching me lecture in person, which is a little bit awkward. But I just met, if you guys are watching this video, I just met you guys for Visio about 20 minutes ago, probably. And we talked through all the class, so hopefully that makes sense. Again, if anything doesn't make sense to you, please email me. My email should be on the syllabus and stuff like that. So make sure you get a hold of me, make sure you guys understand everything. Okay. This class will be fully online, so all the lectures will go this way. I've had success doing this in the past, so hopefully it continues. Okay, so now let's switch to zero. Okay, so to this chapter will be chapter 1, right? So human physiology and integrated approach. Physiology requires that you understand a lot of the anatomy stuff that we went over before. Okay, so if you don't understand or you forgot or it's been too long since you took anatomy, or your anatomy professor didn't explain X, Y, and Z, and that's especially true in the later chapters, since some anatomy instructors didn't really have time to get through all of it. Okay, let me know and I'll find a way to help you, especially since I have all my anatomy lectures recorded already. Maybe you guys can watch those. Maybe we can discuss it, etc., etc. Okay. All right. So here's what we're covering in this chapter. Okay. Okay. So physiology is an integrative science. It integrates. It combines a lot of stuff. Which makes it difficult. Okay, complex systems possess emergent properties, so like you can't understand how a brain works by just studying one neuron. Okay, it doesn't work that way. It only works when you study the brain itself and then try to understand how individual neurons work. So the brain is an emergent property, or brain function is an emergent property of neuron function. Okay, so physiology is all about function, right? Anatomy is all about where things are, where things are located, and that physiology is about how things work. So all about function. Okay, so we will talk much more about function this semester than you guys have probably encountered in previous semesters. Okay, so the definition of physiology, right? The study of normal human functioning. Well, study of functioning of a living organism, but obviously we're going to focus on human. Okay, you don't need to know reptilian physiology, for example, as cool as that is. Okay. So this is all chemistry. This is all molecular biology. This is all cell biology, and then this is physiology. Okay, like my main, so a little bit of background. My main education and my main interest really is on these things right here. I'm a molecular biologist by training. Okay, so in particular genetics. So right here's my main focus. This stuff I've had to take many times because I'm a biologist, and therefore we have to integrate everything together. Okay. All right. So the organ system, make sure you understand what they do. Right. This is like chapter 1 of anatomy. Okay. Okay. So again, this you should review by yourself. Again, this is just like what they do. Okay. 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 One big thing that might help you a lot is mapping. Okay. And I use it all the time. I draw diagrams of how things work. And that actually helps me put things together, especially when it's especially more useful in physiology when you have multiple components that you have to integrate together. Okay, so there's this. A sandwich consists of two parts: the bread, right? The bread, the tortilla, the wraps. Okay, and then you break down the different types of bread and whole wheat and wheat and rye bread and white bread and stuff like that, and flour tortilla versus corn tortilla, et cetera. Okay, but the outside components and the fillings, and then the fillings have subcategories, right? So cheeses, obviously, different types of cheeses, different types of vegetables, et cetera. So every category can have subcategories. Okay, one of the things that's most useful is doing diagrams where boxes are subsets of other boxes. Okay, like for example, celery, carrots, broccoli, cilantro are all vegetables, right? So they would fit into the box vegetables. Okay, and then celery and would go under green vegetables, and lettuce would go under green vegetables. Okay, etc. Okay, so a lot of times when you're studying, one of the things that will be really helpful is to draw these function maps. Okay. Okay. Here is a flow chart. Okay. So person working hot, hot day, you lose water by evaporation. The body fluids become more. Therefore, the body. So every arrow is a therefore. Okay, and you see what happens. Okay. Okay. There's two approaches really to understanding physio, the teleological approach or the mechanistic approach. Okay. We're going to do both. So this is not either or. In fact, things really only make sense when you combine these approaches. The teleological approach is the function explains the why. Like for example, why do blood cells transport oxygen? Because we need all our cells need oxygen. So there must be some way of bringing oxygen to all our cells. In the human body, the red blood cell is that way. Okay. And then there's the mechanistic approach. Well, what is it about the red blood cell that makes it easy to transport oxygen? Okay, the goal is to get oxygen, the red blood cells away. So what's the mechanism? How does it actually work? One, it has hemoglobin. Hemoglobin binds and releases oxygen very easily, so it is ideal for transport. Two, the shape of the red blood cell is a circle. Circles move easier in blood vessels than other shapes. They tend to clog less. Okay, so great. So you have circles. Okay, the red blood cell is biconcave disc. Okay, the biconcave increases the surface area of the cell and allows more oxygen transfer back and forth from the cell. Therefore, they are biconcave. So you can take both approaches. Okay. Okay. Okay, these are the broad themes. Okay, in physiology. Okay, one, structure and function are closely related to each other. Okay, so again, go back to the red blood cell example. Okay, the function of the red blood cell is to transport oxygen. The structure of the red blood cell is a biconcave shape, so it can pick up and deliver oxygen in a very efficient manner. Okay. 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 Lots of concepts. Okay. This chart you can go over on your own. Okay. Okay. So I'm sure if you've been in MSMU for at least a semester, we're talking about the concept of homeostasis. Okay. You've probably heard it before in high school as well. Homeostasis is everything. Okay, illness is now more closely derived as not being in homeostasis. Okay, homeostasis is the maintenance of a relatively stable environment. Okay, so there are critical variables, there are environmental factors and materials, all the factors, all the intercellular communication that has to occur to maintain homeostasis. Like your cells won't know how to maintain heat unless it's told to by the hypothalamus to increase or decrease body temperature. So like your hypothalamus has to measure blood temperature from the outside to figure out what to do with these cells, etc., etc. Okay, okay. Homeostasis is not perfect. Okay, your blood pH is not 7.40000. It is anywhere between 7.45 and 7.35. Okay, so there's a range of values. Your body temperature is not exactly 37 degrees Celsius, right? It will fluctuate between 36.5 and 37.5 throughout the day, and all of that is normal. There's a range of values where things are normal. There's a range outside of that range is not normal, and that causes, or that can eventually cause problems. Not necessarily it will cause problems per se, but it can lead to problems if it stays there too long. Okay. 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 So a highlight to next thing you guys will learn. After you guys learn physio, and I'm assuming all of you guys will pass physio with flying colors. Okay. In physio, we will teach you what happens when things go right. In pathophys, okay, we will teach you what happens when things go wrong. But in pathophys is really important in terms of your nursing career. Okay, yes, I'm talking to you guys like most of you guys are planning to be nurses, which is always the case usually at MSMU. Okay, all right. But pathophys is probably the most important class you guys have to take at MSMU, and therefore physio, you have to understand physio to get through pathophys. Okay. Okay. So homeostasis in a nutshell. Okay. So a organism. Okay. Recording. Okay, so organism always starts off in homeostasis. There is either external change or internal change, but ultimately the external change leads to internal change. Like for example, I take one of you guys and shove you guys into a storage freezer. Okay, that's the external change, right? But being in the freezer will eventually drop your body temperature to where your body can't handle it anymore. So internal change. Okay. Or simply internal change. You run out of blood sugar because you haven't eaten all day. Okay. This internal change will result in the loss of homeostasis. Okay, so you're now outside the range of normal, what your body considers normal. Okay, anytime you're outside that range, your body tries to put it back into that range. Okay, if it succeeds, then you're all happy again. If it doesn't succeed, then you have a problem, and you have an illness or a disease. Okay. Okay. In anatomy, possibly in all the other biology classes you've taken, you probably think there's one internal body environment. That's not actually true. Okay, you have two environments, okay, that are different from each other in significant ways. Okay, one is the intracellular fluid, okay, and that is the fluid inside the cell. Okay, one's the extracellular fluid, and that's the fluid that's outside the cells, that surrounds the cells. Okay, these two are going to be different in critical ways, and we'll talk more about that in later chapters. Okay, for example, sodium will always be outside the cell. Okay, so sodium will always be in the extracellular fluid. Potassium will always be in the intracellular fluid. Okay. Okay. So I love this graph, this diagram. This diagram really works well. Okay. The intracellular fluid is inside the cell, so all the cell surface is the intracellular fluid. It has to interact with the extracellular fluid to pump things in and out of it, and the extracellular fluid has to interact with the external environment. Okay. Okay. For example, okay, so I'm going to be thirsty, so I'm going to drink some water, or in this case, my Genki. Okay. Okay, so when I drink, okay, this is the external environment. Okay, when it enters my stomach, it's still the external environment. Okay, now in my stomach, some of that water is going to be absorbed, okay, into the bloodstream. The bloodstream is the extracellular environment. Okay, the extracellular environment is going to eventually interact with the intercellular fluid, okay, and bring the water from my drink into the intracellular environment. Okay. Okay, the law of mass balance. Okay, so load is the amount of substance in your body. Like for example, your body has 2 pounds of calcium, like a certain amount of muscle, certain amount of fat, certain amount of every chemical, and your body cannot manufacture new stuff on its own. Okay, so there's only so many oxygen atoms that are in your body, and they stay oxygen atoms all throughout. Okay, so you can really only change things a little bit. Okay, so gain and loss. Okay, gain is intake from the outside, or you can make certain things on your own. Like I can use oxygen to make CO2, right? But I can't change oxygen to carbon. Okay, so I can intake from the outside, or I can use metabolic production to make certain compounds, and then the loss is excretion, like sweat or metabolic removal. Okay. Okay. So excretion. So this usually the law of mass balance usually stays roughly the same. So you gain whatever you lose, so that stays the same. Okay. Or roughly the same. Okay. Okay. So there you go. Okay. So usually I drink as much water as I output. Okay. So I could output water by sweating. I could output water by peeing. Okay. Um, and last but not least, I can use water to build new stuff with it, but that's usually a very small fraction of water. Okay. A lot of students confuse these two things. Okay. Homeostasis does not mean equilibrium. Okay, homeostasis is what your body maintains. Equilibrium is the two sides are equal. If your intracellular fluid is in equilibrium with the intracellular fluid, you are dead. Okay, so equilibrium is the two sides being equal. Homeostasis is not. Homeostasis is what your body tries to maintain. For example, all the sodium gets pumped to the extracellular fluid. All right. So homeostasis have disrupt maintain a dynamic steady state. It actively gets into a situation of disequilibrium. The things are not equal. Okay. Okay. So you can see here, this is what I was trying to explain earlier. This is homeostasis. When your body is in homeostasis, all the sodium is in the extracellular fluid, all the potassium is in the intracellular fluid, okay, and all the chloride is in the extracellular fluid, okay, and by all I mean most, not actually all, okay, all right. Okay, so control systems, okay, input signal, integrating center, output signal, response. Okay, I'm sure you guys learned this in anatomy already. Okay, so let's talk about the feedback loops. Okay, feedback loops have a stimulus, a sensor. Okay, input signal, integrating center, output signal, target, and then response. Okay, so a simple feedback loop. Okay, think the knee reflex. The stimulus is the hammer hitting your knee. The sensor will be the receptors on the surface, okay, of your patella tendon that gets the stimulus. The input signal will be the signal traveling through the neuron to the integrating center in your spinal cord. The spinal cord will be the integrating center. Okay, the motor neuron will be the output signal. The target would be the muscles in your leg, and then the response would be the kick. Okay, so feedback response loops. Okay, all right. There are three types of loops. Okay, negative feedback loops, positive feedback loops, and feed forward control loops. Okay, so let's talk about each examples of each one of these. Okay. 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 So, okay, this is a temperature is a negative feedback. Okay, it gets too hot, your body finds a way to cool you down. Okay, it gets too cold, it finds a way to warm you up. Okay, this is actually a heater, but your body works the same way. Your hypothalamus works the same way. If it gets too hot, then you start sweating and you cool down. If it gets too cold, you start shivering, you warm up. And so for a negative feedback loop, you always want to try to go back to something called a set. Okay, your body has set points. Like your temperature is going to be around 37. Okay, your blood glucose level will be around 90 milligrams per deciliter. Okay, etc. So there's always going to be a certain amount of variance, but within that range, you're going to be okay. Anytime you go outside that range, your body's going to react to it and try to bring it back to that range. Okay. Okay. So positive feedback loop. Okay. Positive feedback loops enhance the stimulus. It doesn't go in the opposite direction. It makes it worse. It makes it bigger and bigger and bigger. Okay. So the only way to turn off a feedback loop, a positive feedback loop, is an external factor. Okay. In a positive feedback loop, the response reinforces the stimulus, okay, which sending the variable further away from the set point. Okay, the best example of a positive feedback loop is giving birth. Okay, the baby's head pushes against the cervix. Okay, that triggers the cervix to produce more oxytocin. Oxytocin causes more uterine contractions. The more uterine contractions causes the baby's head bang against the cervix more. More banging, more oxytocin, more contractions, more banging, more like it gets fast. So that's why in pregnancy the contractions get faster and faster and faster and harder and harder and harder because it's a positive feedback. Okay, so the external factor is required to shut off the feedback. The baby's head coming out of the uterus is the outside factor. Once that comes out, there's no more banging, there's no more there's no more oxytocin signaling, there's no more uterine contractions. Okay. So here, this is the positive feedback. Okay. All right. Okay. Sorry about that. Let's see. Okay, so positive feedback loop. You guys can watch this on your own time later. Okay. Well, circadian rhythms. Okay. So circadian rhythm, you have a feedback loop. Okay, so you normally respond to this. Okay, your temperature is usually warmer in the dark, which is why fevers usually occur at night. So in reality, what actually happens is you notice the fever at night because you're warmer and therefore even a little bit of it is more noticeable. Okay. Okay. One of the things that we want to make sure you guys understand in physiology, and this will be more explained in the labs, hopefully, is this kind of idea of how science actually works. Okay. Okay. So there is a concept of independent versus dependent variables. Okay. Independent variables are what you as the researcher decide to do. Like the groups that you decide to put things in. The dependent variables are what you actually measure. Okay. You always need good experimental controls, and hopefully your instructor will talk to you about that. Your lab instructor. Okay. You need data, you need good data, and you need replication. Okay, you can do something once. Okay, you try to do something once, and you try to tell somebody about it. Most science people, including myself, will laugh you out of the room. You need to do things many times, many different kind of replications, and to justify your hypothesis. Okay. All right. Okay. So when you graph, the independent variable is always going to be a horizontal axis. The dependent variable, what you're measuring, will be on the y-axis. Okay. Bar graphs, line graphs, and scatter plots. Bar graphs are when the independent variables are distinct entities. Okay. So you have one group taking the medicine, you have one group taking the placebo. These two, like, there's no overlap between the two groups. There's not one person taking both. Okay, so you're either in group A or you're in group B. Those are bar graphs. Line graph are the independent variable is a continuous phenomenon, like your weight over time, or the temperature of the room over time. Okay. Scatter plots show the relationship between two variables, like the number of hours studied versus your grades. So everybody has the number of hours they studied and grade. And and so there's like multiple data points. I'm gonna graph all those data points, and then I can see a relationship, hopefully a positive correlation between the number of hours you studied and grade. Okay. So this is a scatter plot. So, okay. Okay. So this is especially true. So this is more for lab that will actually make you draw graphs. So make sure you do this right. Graphs will always be dependent variables, independent variables, okay, and in units. Never ever forget units. You forget units, your entire graph is meaningless. Okay. People have shown me graphs without units before. I just gotta go. Yeah, that's BS because there's no units. I don't know what the hell you're measuring. So make sure you include titles here. So a dependent variable, an independent variable, and also a title for the chart itself. Okay. Usually the title should be the effects of X on Y. Like, so you're always telling what you measure. Okay. Bar graphs, okay, line graphs, scatter plot. Okay. All right. Okay, so we will go over these guys in the lab. If you guys don't, I'll find some time to go over this with you guys. Okay, all right. So that ends chapter 1. So hopefully you guys learn something. If you guys and what will happen is we will go over this in class first thing the second time we meet. Okay, all right. Bye everyone.